This is Audiommunity, a podcast about our body's never-ending fight with the outside world.
You get a chance to see in the paper some of the effects of those drugs. So some of their patients have some complications from immune suppression. I think like one or two of them get disseminated HPV, which sounds awful.
The rules of inheritance that you probably all learned in high school biology don't really work for MHC, but they don't know that they don't work.
The models that we're going to be talking about today involve taking a solid piece of biological tissue from one organism, putting it into another, and that doesn't work, usually. Neither of us are going to die from each other's tumor, which is good.
Why are we transplanting your tumors to your brothers?
Welcome to episode 1 of Audiommunity. I'm Kate, I'm a graduate student in virology, and joining me today is Matt.
Hi everybody, I'm Matt. I'm also a graduate student. I am in the immunology program, however, so we are a warring faction with the virologists.
Also joining Matt and I today is another graduate student, Kevin.
Hello, hello. I'm also in immunology.
Today, our podcast is focusing on transplantation. And what we're talking about is organs and bone marrow transplants, which you may have heard about on the news before. And likely what you've heard are the complications that occur from transplantation. And it's often very hard for someone who's in need of a transplant to find a proper donor. And so why is that? The problems with the donor do not necessarily arise from the functioning of the organ, but instead it's actually a very important immune system problem. We will be discussing some of the underpinnings of this problem. Today, specifically, we'll be talking about some interesting papers that talk about the advancements in graft acceptance and a historical view that defines one of the main mechanisms that govern not just why transplant may be accepted or rejected, but it's also a major concept in immunology that relates to how the immune system can see pathogens, see itself, and tell the difference between those. And from this, we start to understand how an immune response is actually developed. We're going to start off with a discussion of a historical paper that really starts to define the differences between MHC and their role in transplantation. Matt, why don't you start us off?
Sure, so let's talk about the paper. The paper itself is called Alleles at the Histocompatibility 2 Locus in the Mouse as Determined by Tumor Transplantation. So that is by George Snell and George F. Higgins, both Georges. But before we really get into that paper, I think it's important to talk about some of the really basic background stuff that we'll probably be discussing a lot over the next several minutes. I don't know how long this will actually go. We should talk about MHC, which is really going to form the basis of how a lot of this works. And probably just a little tiny bit about T-cell responses and what MHC is used for when we're not talking about transplantation. So for the most part here, the models that we're going to be talking about today involve taking a solid piece of biological tissue from one organism, putting it into another. And that doesn't work, usually. It's not an easy thing to do. And the reason that it doesn't work is because we all have very, very different types of this molecule called MHC. And so the question is, I guess to start, what is MHC? Before you really understand exactly what MHC is, you have to understand that there's a major component of our immune system. They're called T cells. And T cells are responsible for a lot of the functions of what people consider sort of fundamental hallmarks of the immune function. Finding cells that are infected with viruses and killing them, or helping other cells produce antibody. All these things are done by T cells. But T cells are not capable of just seeing a bug, seeing a virus, seeing a bacteria. They can't just see those things and start responding against them. What they need is some sort of help. And that help comes from something that we like to call an antigen-presenting cell. The antigen-presenting cell will find the bug, say, I don't think this really belongs here. take that bug, cut it into little tiny pieces, and show it to the T cell. Why is that important for MHC? It's important for MHC because MHC is actually the molecule that antigen-presenting cell will use to actually physically show the T cell that piece of the bug.
Let me just jump in here and say, this is in contrast to antibodies, which detect things on the outside of cells. Some pathogens infect inside of cells, and antibodies can't reach them inside of cells. So it's important to have a way for cells to display what's happening on the inside to the cells of the immune system in order to respond to those what we call intracellular pathogens.
Yeah. So these are ways that we can detect that there's actively replicating virus, which is really important because, you know, you might not be able to stop all the virus that's just in the bloodstream, but if you can find cells that are producing viruses through like the presentation of the viral peptides on the MHC, you can kill them, which is what the T cell does, and start to clear the infection that way.
Yeah, and also Matt mentioned that the antigen-presenting cells know that something's wrong and present that bit on the cell surface, and that's partially true, but actually antigen-presenting cells are presenting all of the bits on their inside to T cells, but T cells are only going to respond to the foreign bits, or at least they should only respond to the foreign bits, and that's going to become important later on in our discussion of transplantation.
Right. And we'll talk a lot about T cells, by the way, over not only this podcast probably, but other podcasts as well. And the truth is that there is a huge variety in the numbers and kinds of T cells that we have. They're all slightly doing different things. They're all responsible for different parts of immunology. But for now, why don't we just think about them in the context of transplantation as these really stupid killers? They're not actually capable of discerning who's good and who's bad on their own. They need somebody else to come in and sort of say, hey, that guy over there, he sucks. Right? We need somebody to go take him out. You have these T cells and they're wandering around and they're looking for an antigen presenting cell to show them something and say, you know, this is something to be aware of and it's a problem, so go kill it. And so why is MHC important? MHC is critical in that actual interaction between the APC, the antigen presenting cell, and the T cell itself. So the MHC is going to be the molecule that the APC uses to actually present the antigen to the T cell. You can think of it almost like a flagpole. So the flagpole itself doesn't actually tell you anything. It's the flag that tells you something. So MHC is sort of akin to the flagpole in this situation. So when an antigen-presenting cell finds a bacteria or a virus, it sort of turns that bacteria or a virus into a flag, and it runs that flag up the flagpole. So it basically presents the piece of that bacteria on its MHC, and it shows it to the T cell. So why do we have such diversity then in MHC if you're just trying to present something to a T cell? And the reason is that all of us are susceptible to infection. That's not really news to anybody, I guess. But certain MHC molecules are much better than others at presenting certain pieces of infectious material. So certain viruses and certain bacteria fit better onto different kinds of MHC than the others. And the reason that's important is because almost all of the susceptibility, so, you know, I grew up in a house of 3 boys, so me and 2 brothers, and then my parents, and we all would usually get the same diseases, but not always. Every once in a while, somebody would be spared. The reason for that is because we all have our own repertoires of MHC, and almost 100% of the variability as to whether one person is going to get sick from a bug and the other people don't is because I probably have a better or a worse MHC at presenting that bug than you do. So MHC is hugely important in controlling immune activity in the normal human population, and it's really important that it's diverse so that a single virus can't come through and just wipe out the entire human population, right? If we're all genetically identical, we can all fight the same virus at the same capacity, then something comes along to subvert that, and all of a sudden we're all dead.
Yeah, it's worth noting in your example that bad MHC for a particular viral infection is only bad for that viral infection. It's not like there's bad MHC and good MHC.
Right.
There are bad MHC for particular viruses or in other infections, and there are good MHC for particular bacteria and viral infections.
Right.
So that MHC that's terrible at combating this year's influenza might be great at presenting stuff from last year's influenza or from rhinovirus or something like that.
Yeah. And so you're absolutely right. There is no such thing as a good or a bad MHC unless it doesn't do its job. But what is good, undeniably, is diversity in MHC. So when you don't have diversity, the whole population becomes susceptible to specific viruses, bacteria, and there are tons of examples of this, right? So banana trees are genetically identical, which is why there's so much concern over the fact that all the bananas are going to die. We're never going to eat bananas again.
Well, at least not Cavendish bananas.
Is that like Cavendish?
What now?
What's a Cavendish banana? Like Cavendish Labs
It's
banana?
the only banana that anyone has ever seen. Cavendish is the strain of banana that we eat in this country.
I did not know that.
Does it come from Cavendish Labs?
I don't believe so, although...
That seems like it would be hard.
I am doing this right now. I'm Googling this. Cavendish.
I'm sure Cavendish was a very wealthy family in Britain or something, and they just happened to name the banana as well as the lab.
Oh, okay. I was going to say, because it's also where, like, the structure of DNA was uncovered.
That will be an interesting segue to the future of this conversation. So yeah, when you have these genetically identical populations, they're incredibly susceptible to single pathogens coming through and wiping virtually everything out. And that's what's happened with bananas. It's also, by the way, why you don't allow animal populations to dwindle down to what we call the bottleneck level. Because as soon as you bottleneck genetic diversity down enough, it means that the MHC isn't varied enough, which means that, yes, you can have 100 cheetahs in the world, and they can propagate forever, and they can be super great breeding cheetahs, and that's all fine, except that you've lost all the diversity in the MHC, so as soon as the first cheetah virus comes around, they're all going to get killed by it anyway. So even if you repopulate the whole world with cheetahs, they're all going to die. By the way, I'd like to point out that has not happened. That has not happened with the cheetah population yet. But
it's only a matter of time.
For those of you who are environmentalists, this is why you should be out there working harder. So, anyway, now that I've offended the entire listening audience, why don't we get back to this paper? So, if we talk about the idea of MHC diversity and how important it is in the immune system, you can imagine that your immune system can recognize your own MHC. So when T cells develop, they develop in such a way where they're actually instructed by your MHC. Basically, they're shown the flagpole and they're told, this is what the flagpole looks like. And what you're looking for is the flag. And so then they go out into your body and they see all the different flagpoles and they wait until they see the flag that they recognize. Now, when that's different, when the flagpole doesn't look right, T cells have a tendency to freak out because, like I said, they're sort of dumb killers. They don't really know what to do. So if they haven't been specifically instructed not to kill the flagpole, then they're going to. So this is the fundamental basis of what we're talking about when we're talking about transplant rejection. You have a T cell that hasn't seen a different kind of MHC than the ones that it grew up with, and as a result of that, it just sort of freaks out and kills everything. So in the case of transplant rejection, and in the cases that I'll be talking about with this paper, what we're talking about are tumors. And so everybody thinks about tumors as, cancer. Obviously, they are. Tumors are naturally occurring, but you can imagine that if you take a tumor from one person and you take it out and you put it into another person, it actually won't grow. And the reason that it won't grow is because the person who receives the tumor, their immune system is going to see that tumor and say, hey, look, the MHC is completely different. And if the MHC is different, this is not a part of us. And if it's not a part of us, it has to go. So this is tumor rejection. And so this forms sort of the basis of the paper that I just mentioned by George Snell.
Although it's worth noting that while in humans, tumors normally can't be transmitted, there are actually natural examples where MHC diversity is really low in a population, And there are infectious tumors, and I'm thinking specifically of Tasmanian devils, which have this horrifying facial tumor disease that's spread when Tasmanian devils fight each other during mating, of all things. So it's kind of a sexually transmitted disease, but it's a tumor that started in one Tasmanian devil that is now propagating throughout the population of Tasmanian devils, and their immune systems can't fight it because the MHC diversity in those populations is so low. So we don't have to worry about infectious tumors, thankfully.
So to console our listeners, you will never be subjected to a sexually transmitted, horrifying face tumor. That's a good start, I think, to this discussion.
I kind of want to put like an addendum on that, unless it's like a virus tumor, in which there are people who get virally induced cancers that could affect their skin. And I imagine they could be transmitted sexually if you are rubbing your warts together.
I feel like this conversation has just taken a turn, and I'm going to go ahead and...
But I mean, I didn't want to put them like...
and direct us... Let's direct us back. Yeah, this is what virology does to you. Virology.
I did
not
want to
even
say,
once.
Though, I figured out the 7 or 6 degrees of Kevin Bacon between the banana and Cavendish laboratories. Because as you were talking, I was furiously Wikipediaing. The Cavendish banana is named for William Cavendish, the 6th Earl of Devonshire, something like that. And his...
I was going with 5th.
Yeah, 6th. I know, it's tricky. And then his nephew, his sister's son, became the 7th Duke. Did I say Earl? I meant Duke. The 7th Duke of Devonshire. And his...
Let's get it straight, Kate.
Yeah, I know,
I'd like
exactly.
to know what
Don't
the
you know the
difference
degrees
after
of the
this.
peerage?
Yeah,
And his
exactly.
relative is Henry Cavendish. The laboratory is named for Henry Cavendish, who was a chemist, and his relative is the 7th Duke of Devonshire, who was also the Chancellor of the University of Cambridge in England, and that's where Cavendish Laboratory is. So it's named after Henry Cavendish / the 7th Duke, who is, obviously related to the 6th Duke, who was a huge, plant guy.
So if you're a Cavendish out there and you would like to fund this program, please get in contact with Kevin, because we're apparently going to plug your entire family tree. We'll just keep going, I promise. Our whole next episode will be about the Cavendishes and their input into science. It'll be wonderful.
29 Cavendish researchers have won Nobel Prizes. That has to be a record.
That doesn't even make sense. None of that doesn't make sense.
No, not like people who research Cavendish ancestry, but like the actual labs. Maybe I should have.
Okay.
Maybe
I thought you meant the family, the Cavendishes, had won 20. That would be a lot. That would be hardcore Nobel.
I bet there's
that's
3
lineage
though.
right there.
There's probably one. Maybe there's one Cavendish, but
all right. Well, let's assess that at some point. And so really, what this older paper starts to lay out is exactly what this genetic diversity looks like. As I mentioned, someone is unlikely to be able to transplant their own tumor to someone even closely related to them, because genetic diversity, MHC diversity, between even me and my brothers is going to be really, really high. So my brother is going to recognize my tumor as foreign. I'm going to recognize his tumor as foreign. Neither of us are going to die from each other's tumor, which is good.
Why are we transplanting your tumors to your brothers?
Well, because I used the brothers as an example earlier as how we, probably would get sick from each other, but not always, right? So we definitely wouldn't be able to transplant tumors because even small variations in MHC are recognized heavily by your immune system as foreign. So this is really the focus of this paper, is that you can imagine that if my brothers and I were genetically identical with each other, if my tumor went into my theoretically identical twin brother, that would actually work because my immune system and his immune system would be trained on the same MHC repertoire. And as a result, when my tumor went into him, he wouldn't be able to recognize it as any different from his own body. And so through the power of inbreeding, We have actually been able to create scenarios like that in mice, and this is where this paper really starts. So when we talk about inbred mice, all we're really talking about is mice that have a litter, and then a brother and a sister from that litter are crossed into each other, and then a brother and a sister from that litter are crossed back. And I think, what's the number? It's something like 8 times for rough genetic purity. Do you remember the number, Kevin?
I don't remember exactly. I thought it was more than that.
Well, so I thought it was 8 to get like a huge percentage, and then in order to be really sure, I think the number is actually like 20 or something like that. In any case, when you backcross mice enough, they become genetically identical to each other. And so all of the siblings of a specific cross look exactly the same as the parents and their grandparents and all of those. And what that really means at the molecular level is that the offspring are going to have genetically identical MHC as the parents, which means that if you take a tumor from the parents and you give them to the offspring, or if you take a tumor from one of the offspring and give it to another offspring, they won't recognize it as different. So your immune system will see it and say, hey, that's the same flagpole that I'm used to. Doesn't matter what's on it because we're all good here. So this is how tumor transplantation works. And this is how they started to really understand what MHC was and what histocompatibility was. Histo, of course, meaning tissue, and then whether or not it's compatible in another organism or not. And so really what this paper is focused on is looking at different genetic strains. So if I take mouse A and B and mouse C and D and I cross them, and then I start back crossing them and back crossing them and back crossing them, eventually what I'll find is I'll have 2 distinct strains. They will both be genetically identical to their offspring, but they won't be genetically identical to each other. So if I take a tumor from genetically pure mouse strain A and put it into genetically pure mouse strain B, it's likely that I've selected for very different MHC markers along that crossing system. And so this is where this paper is going. It's taking a look at some of the really commonly used back-crossed strains that were already available at the time. But the idea here is trying to investigate which mouse strains have the same MHC, which ones have different MHCs, and we're going to do that by trying to just transplant tumors from one mouse strain into another and seeing if the tumor grows. If the tumor grows, then it means that those 2 mice must have had exactly identical MHC because the recipient of the tumor wasn't able to fight it off. And so one of the important things to really note about this paper is that the timing of this is actually quite interesting. So this paper was published in 1950. Actually, it says received August 29th, 1950. It's really important to point out that Watson and Crick did not publish the structure of DNA until 1953. That's important because basically what we're talking about in the Snell paper is a system where people had already figured out inheritability. So all the way back to Mendel, we knew that you could take shriveled up peas and they would transmit to their offspring, right? So the idea of inheritability was already there. It had sort of been rediscovered and it was being worked out in mammals in the early 1900s, but it wasn't until 1953 with the publication of the structured DNA that we actually knew how DNA might be able to replicate. And that's important because when Snell was doing this work, we're talking about the inheritability of MHC genes from parent to offspring, but you don't actually know how that inheritance works yet. You don't know how the DNA is being transmitted from parent to offspring. And in some cases, people didn't even know if it was really the DNA that was coding for these MHC regions. So one of the reasons that this paper is so interesting is because we're talking about trying to work out a system where, for some reason, tumors are capable of being transplanted between some mice, they're not capable of being transplanted between different mice, and you're trying to figure out why without the added knowledge of how things are inherited from each other even.
Yeah, the rules of inheritance are pretty clear. People have been working on that for a long time, but the molecular knowledge that we've gained in the last 60 years is, completely opaque to them. And it's interesting to note that the rules of inheritance that you probably all learned in high school biology of dominant and recessive, maybe you remember the terms codominant, don't really work for MHC, but they don't know that they don't work. So those terms all refer to things that are having effects and they're at a single genetic locus. MHC, we now know, is actually multiple different genes that are all really close together. So they tend to be inherited together, but they don't always get inherited together. And the way that dominance works is that if any one of those MHCs are different, that will confer dominance. So it's this really weird mechanism of inheritance that had never been discovered before or had never been known about before. And these guys are doing these experiments and they're able to reason out a little bit of what's going on despite these huge fundamental gaps in knowledge.
Yeah. And so this, Kevin just sort of hit on why this paper is actually very complex. I had a hard time reading it now, and I know exactly how inheritance works. I know exactly how MHC works, and this is still a difficult paper to understand because genetically it's actually a very complicated question that they're asking. And they're asking, what is it that confers acceptance of a tumor transplant? And when you see a mouse that rejects a tumor, what really is going on? And as Kevin mentioned, the assumption is that there is a single gene that's different between mouse 1 and mouse 2. And when mouse 2 sees the difference in that gene, it's going to reject the tumor. And so they set up all of these really complicated genetic crosses to figure out what kinds of MHC all of these different mouse strains had. But in the end, MHC is not a single gene. MHC in humans is actually 6 genes, a little bit less than that in mice. It's still 4, though, that are coding the major types of MHC. But at the very least, you're talking about 4 independent genes that are all responsible for the same sorts of outcomes. So while they were trying to track differences in one specific gene, they were really accidentally looking at 4. They're trying to figure out all of these really nuanced concepts in a time where you didn't quite yet understand how DNA worked. Now, you did understand a little bit that tissue could be transplanted. So, way back, there are early reports of people trying to transplant tissue in early Indian culture. There are reports of transplantation, some really early Chinese accounts. It obviously never worked particularly well because no one quite understood what they were doing. But by 1916, people already understood that tumors could be transplanted between inbred mice. So the idea of histocompatibility had sort of been born. And by 1927, the first transplants in humans had really been documented, where identical twins, it was found, could transplant skin grafts back and forth. So they understood that there was something that was genetically distinct between humans, but the complexity of that sort of escaped them, and necessarily so. It wasn't as if they were missing something. It was just that the information was very complicated and difficult to get at. So the one thing that becomes really difficult in papers like this is often the terminology that's used. In this case, the terminology is largely H2. They talk about the H2 locus. And if you read the paper, it becomes clear that when they say H2, what they're really talking about is MHC. But H2 is just the term that's used for mouse MHC. And so in this paper, the first author, George Snell, had already mapped out 3 different kinds of genes at the H2 locus. That's what he was calling it. There was a guy, Gorer, P.A. Gorer, who actually had joined Snell a while back, and Gorer had been identifying different kinds of blood antigens that made my cell different from your cell. So he had found 4 potential differences between cell types that would explain the difference that your immune system sees. It turns out that the second of 4 that he identified was the MHC locus. And at the same time, Snell had been looking at histocompatibility genes. That's what he was calling them at the time. He sort of converged on the same locus, so he sort of independently started to discover MHC. And the result of that was they basically just smashed their names together when they were both working at Jackson Laboratories later in the 40s, and so it became the histocompatibility 2 locus. So when you see H2, it's really just referring to the MHC locus. And the reason that that's important is that it would eventually become known that there were multiple genes, multiple different MHCs at the H2 locus. Like I said before, in mice there are 4. So a lot of the naming systems became really messed up. So when you read through the paper here, you've got H2, which is the gene itself, and then you get things like H2A, H2B, H2D, and those H2 designations, the superscript designations, basically said, well, we know that they have an H2 locus, but it's different from the one that we previously identified because these mice reject the tumors from the other mice. So instead of calling it just H2, we're going to call it H2A and H2B now.
So those are different versions of the same gene.
Right, and we call those versions of the gene alleles. So it was discovered that there were different alleles at the H2 locus. The problem with that, of course, is that when you realize that there are 4 genes in that locus rather than just the one, all of a sudden you're stuck with that superscript. So if in black 6 mice you had named it H2B, so you said it was allele B at the H2 locus, when you realize that there are actually 4 genes, now all of a sudden you're stuck with that B superscript. So instead of just H2B, it's now H2K superscript B, IAB, IEB, and H2DB.
And this is the bane of immunology students everywhere.
Exactly. So this is why we all tried to jump out the window when we learned about MHC, is because the nomenclature here is stuck in the history. And it got really complicated when it was realized that even though the DBA mouse, which is just another back-crossed mouse, and the A mouse couldn't receive tumors from each other, so they are MHC distinct from each other, some of the MHC alleles were actually the same, but it was the ones that were different that was causing the tumor rejection, which means that even though the A strain mouse started as H2A, all of a sudden you have 4 genes now, and it's H2KK, IAK, IEK, but H2DD. So some of these mice strains and the way that we named them really got in the way of even understanding how all of these things work. So if you're ever diving into MHC diversity, honestly, you have a huge task in front of you. And this is in mice. You know, we only have a couple of mouse strains that we really use all the time. If we're talking about humans, you're looking at 6 genes, of which there are between 1,500 roughly and 8,000 different alleles at each one of those MHC genes. So if this was ever a plug for joining your bone marrow registry, I guess this is it, because the chances of you finding someone who is similar to you at the MHC locus is slim to none.
And if you tuned out at all for the discussion of MHC nomenclature and naming, this would be a good place to jump back in.
Yeah, exactly.
MHC is really, really diverse.
It's super diverse. And this is actually a really good segue to the second paper. Not to not go into the specifics of this one, but let's be honest, it's really complicated and the genetics are sort of boring. So what is MHC important for? And Kevin will be describing the newer paper, talking about how you sort of use tricks, immunological tricks, to get around the fact that while me and my brother can't give us tissue, maybe if you outsmart the immune system a little bit, we can.
Right. So the second paper we're going to be talking about is a paper that was published in 2008 in the New England Journal of Medicine, and it's called HLA, Mismatched Renal Transplantation Without Maintenance Immunosuppression. And so the background for this paper, first of all, HLA is just the human lymphocyte antigen, which is just human MHC. So all that stuff Matt was talking about with H2, that's mouse MHC. HLA is human MHC.
You're welcome, everyone who's not an immunologist.
Yeah, exactly. And as Matt mentioned, human MHC, or HLA, is super diverse. And there are tons of different versions, tons of different alleles at these 6 different places in the genome. And this makes finding a match for transplantation really difficult. And unless you have an identical twin, there's almost no chance of finding an HLA identical donor for any sort of transplant. And so usually what this necessitates is the use of tons of immunosuppressive drugs for transplant recipients. So if you get a kidney transplant or a liver transplant from someone that is HLA mismatched or MHC mismatched, you're going to need to be on drugs that tamp down your immune system basically for the rest of your life. And this kind of sucks.
It should be pointed out that these drugs, I mean, we talk about how they suppress the immune system, and so one might anticipate that you're going to get infections more often, and that is certainly true. But outside of that, and probably even more importantly, these drugs are really, really nasty. They don't do good things to people, and they have been lifesavers. They have been incredibly important in allowing people to receive transplants, in controlling autoimmunity. But should make no mistake that these drugs are quite nasty despite their life-saving effects.
You get a chance to see in the paper some of the effects of those drugs. So some of their patients, when they go through their patient history, they have some complications from immune suppression. I think like 1 or 2 of them get disseminated HPV, which sounds awful.
Yeah, HPV is human papillomavirus, which causes warts. Fun. Throughout your body.
Nobody likes warts.
And it's also the causative agent of cervical cancer and possibly non-melanoma skin cancers. But that's one of those things. That's like some of the risks that you take when you're on immunosuppressive drugs.
Right.
That you no longer can keep these infections in check like you would if you're healthy.
Yeah, so those drugs really suck, but they're better than kidney failure. So the goal of this paper is to figure out a way to do kidney transplants without the need for constantly being on these terrible immunosuppressive drugs. And in order to do that, the authors use our understanding of how the immune system works and how it develops in order to try a procedure that is actually pretty impressive. So before you read this paper, just think about the background. That is, it's impossible to find matched donors for organ transplants unless you have an identical twin. And if you don't have MHC matched donors, you're going to need to be on lifelong immunosuppressives. And even then, the graft might not survive for very long. So a lot of people that get organ transplants actually need to get repeated organ transplants because eventually, even with the immunosuppressive drugs, the immune system is eventually going to figure out that there's something going on there and reject the graft. So in this paper, what they're doing is they're taking advantage of our knowledge of how the immune system develops and how T cells that Matt was talking about earlier learn to recognize the flagpoles and the flags. That happens in an organ that's called the thymus, that's sort of sitting above your heart in your neck region. And in the thymus, new T cells, as they're developing, learn to recognize their own MHC. So T cells go through the thymus and they're looking around. If they detect MHC really strongly and bind really tightly in a way that might activate them in the future, in the thymus, they're killed off because the immune system recognizes that if T cells are seeing something really strongly in the thymus, then they must be reacting against themselves, and that's a really terrible thing. So they get deleted.
Right, and this is the start of autoimmunity under some models. You have a T cell that recognizes your own MHC as bad. It reacts, and so you have cell death and you have basically the start of an autoimmune response. So really what we're talking about here, it's called central tolerance, and it's a way to prevent your own T cells from causing problems in your body later on.
Right. And under normal conditions, when someone gets a solid organ transplant, like a kidney transplant in this case, they have T cells that developed looking for their own MHC. So any T cells that recognize the recipient's MHC would have been deleted in the thymus, but a lot of those T cells might recognize some other MHC that's present on a donor's tissue, and they wouldn't have been deleted in the thymus. So when they run into that kidney as they're perusing around the body looking for infection, they're going to see that MHC bind tightly and think, oh, these are infected cells. I should probably kill that.
I like thinking of perusing T cells with sunglasses, by the way.
So what these authors decided to do was to try to educate the recipient's T cells to the donor's MHC. And the way that they do this is, in addition to giving them a kidney transplant from the exact same donor, they're going to give these recipients bone marrow transplants. And bone marrow is the source of stem cells for the development of the entire immune system. Some of those bone marrow cells are going to develop into those cells Matt mentioned earlier, the antigen-presenting cells, and they're going to go to the thymus, where they can educate new T cells and teach them that this is self-MHC. Basically, you're going to trick the recipient's immune system into thinking that the donor's MHC is self-MHC. So in this paper, they describe 5 different patients that they tried this new procedure on and got pretty remarkable results, considering that before this, all transplant recipients had to be on lifelong immunosuppressive drugs. These 5 patients, or at least 4 out of the 5 patients, managed to go off immunosuppressive drugs entirely, and their grafts survived for several years at the time of this study. So I'm not going to go into a ton of the details of this paper, but the important thing to note is that this is an example of using our knowledge about the immune system to improve outcomes in human diseases. And so 4 out of these 5 patients managed to go off immunosuppressive drugs after about a year, and then had their grafts survived for, in some cases, several years. And actually, there was a paper that was published just this month that talks about the long-term outcomes of this study, and they've actually tried this procedure on a number of other patients. I think they're up to 10 now, and they have 8 out of 10 of these patients still surviving without immunosuppressive drugs, and their graft still hasn't been rejected, which is pretty remarkable. Some of the patients developed some graft rejection. Some of them had to go on minor immunosuppressive drugs after several years of being off immunosuppressive drugs, which is unfortunate, but it's still a huge improvement over what was going on before.
So an interesting thing, I was actually talking with someone, one of my roommates all of my roommates are immunologists, and one of them went to an international conference on transplantation. I was talking to him about this paper in preparation for this talk, and he mentioned to me that at the conference, it was recognized that this had come about, but no one was really talking about it. You know, it was sort of understood that this really cool thing had been done, this outcome had been reached, but the transplantation field as a whole wasn't really discussing it all that much. And it's surprising to me, I suppose. The impact of this paper to me seems to be huge, honestly.
Yeah, this paper was published right when Matt and I started in the immunology program. So it was a brand new thing and super exciting at the time.
Yeah, David Sachs, I believe, is the last author, correct?
Yes.
So David Sachs is actually a faculty member within our program, and I was very excited at the possibility of working with this guy because this is the sort of thing that really changes huge amounts of people's lives. The prospect of transplanting kidneys or skin or, anything else you can think of, brains. You can't actually transplant brains. So the prospect of being able to do this sort of freely, to move organs between people, is huge. There's a huge shortage of organs out there. Even if you do this completely, there's still going to be a huge shortage of organs out there. But right now we're in a situation where for every organ transplanted in this country and around the world, you also have one person that is going to have to be on immunosuppressives for the rest of their lives. So, it doesn't take much thought to really sort of extrapolate the impact of that.
And going beyond even human transplants, people have talked about, using animals to grow organs. So, for instance, apparently pig hearts are about the same size as human hearts. So people have talked about genetically engineering pigs so that you could use their hearts for human transplants, but you're still going to have examples where the immune system recognizes the difference between that organ and their own organ. And if you could have a way of training the recipient's immune system to accept a transplant, even if it's not genetically identical, that could have huge implications, which could be a really wonderful avenue for therapy.
What I wanted to say about Matt's comment about how this procedure hasn't been picked up in hospitals and in medicine today. None of us are doctors, and I wonder if the idea of doing 2 transplants at once is somewhat daunting. There's some risk associated with these that...
Yeah, so one risk that I didn't mention is anytime you do a bone marrow transplant, you have the risk of graft versus host disease, which is sort of exactly what it sounds like. Bone marrow transplants, because they contain immune cells, sometimes the graft bone marrow can actually develop cells that attack the recipient and cause really terrible disease, GVHD, graft versus host disease. In this procedure, they actually didn't see any graft versus host disease, and the donor bone marrow transplant only stuck around for a little bit. Actually, it went away over the course of the first year, and they didn't see any noticeable graft versus host disease. And I think that was true in the follow-up paper that I read that came out just this month as well. But anytime you're doing a bone marrow transplant, you also have to knock down the recipient's bone marrow a little bit to make room for the donor bone marrow. So there might be some problems with that. But based on these publications anyway, and you're right, none of us are doctors yet. I'm going to be hopefully soon, but not that kind of doctor.
But not the kind that helps people.
Not the real doctor. Not the sort of doctor that can evaluate medical procedures.
Right. But it seems like based on this paper, the upside of being able to go even for a few years without immunosuppressive drugs seems like a pretty good compromise. And there are almost certainly risks involved, but there's risks anytime you get a transplant. And it seems like until we start noticing real problems with this procedure that we should be trying it more often.
So I'd like to point out that I actually asked this question. Why is it, do you think, that people are sort of keeping this off to the side a little bit? And I think part of the reason, at least in my understanding, seemed to be that people don't quite understand it. People don't quite understand exactly how it works. And to be honest, this is an extremely complex topic. It was ambitious of us to try to, first of all, read and understand, but also to explain exactly how this works in our first episode, because this is something that is relatively new to human health. Transplantation, I said that it's been around for thousands of years, the record, but realistically, transplantation has been around since the mid to late 60s. We have only been doing this for 50 years, and much less than that effectively. We're talking about an immunological concept that is incredibly complicated. It's known how grafts get rejected, but I would say, prior to the taping of this, Kevin and I had a discussion about exactly how it is that transplants do get rejected, exactly how the recipient's T cells recognize the donor's MHC. And the true answer is that it's not 100% clear.
And it should also be noted that I mentioned part of the reason that we think this works, that some of the bone marrow cells with the donor MHC are going to the thymus. But there are also a whole host of T cells that have already developed in the recipient that don't have to go back through the thymus. And somehow this procedure is preventing those cells from being reactive against the graft as well. And there could be something going on with T regulatory cells. There could be a number of other mechanisms that are going on. It works, but we actually don't know exactly why this procedure works.
Yeah, they try to get more into the mechanism in the paper with some very light in vitro studies. But I think as an actual rigorously tested hypothesis, they don't really come to any conclusions with their own figures, but just more suggestions as to what is actually occurring and what is inducing tolerance, becoming non-reactive, tolerating the graft. It's just more hinted at in figure 2 than it is actually proven.
Yeah, there are a number of plausible ways that this could be working, but they don't actually know what is the way that it's working.
Right. So maybe the right move, instead of piggybacking everybody onto this study, is to sort of use this as a benchmark. This is a good way to, as Kevin mentioned, use sort of our understanding of how immunological tolerance develops in a normal host, and then hopefully start to apply that to transplants and to places in human health where it could really be of use to people.
Yeah, though I do also want to mention that we have a long and glorious history in medicine of doing things even though we have no idea how they work, just because they work.
Yeah.
Yeah, even if we don't understand exactly how this procedure works to generate immune responses that prevent transplant rejection, it's still a really cool example of taking experimental knowledge about the immune system and translating into a therapy that seems to work, even if we don't know exactly how it works. So that's pretty cool.
And hopefully we'll follow that up and we'll know exactly how it works in the next 10 years, and then everybody can get everybody else's kidney whenever they want.
We can only hope.
That's the future, according to Audiommunity.
All right. So this has been Audiommunity from eMunity.org. I'm Kevin Bonham with Kate Franz and Matt Woodruff. You can find us on SoundCloud at soundcloud.com/Audiommunity, and eventually we're going to be on iTunes, so look for us there. You can find me on Google Plus and Twitter. We have a Facebook page, facebook.com/Audiommunity, and all of these links and email addresses for the three of us can be found at our website, emmunity.org. We're supported by Harvard Med School, and the music at the beginning and end of this podcast was composed by Rachel Reineck. Thanks for listening. We'll see you next time.